All right. So, hang on a second. Okay, wrong button. Doing this so I know what we're doing. Okay. I'm going to try to leave the lights up unless you tell me that you cannot see it, and then I will turn them down. But that way you can see other things if you wish to. So first one, it says, can describe an intracellular versus extracellular electrode, and then speculate on the advantages or disadvantages each of those of the two techniques. And it does say speculate because it doesn't really tell you much, but at least you could think about it. So who this go? This one to Leandra. So what'd you get? Did you got anything for advantages, disadvantages? That's okay. Yeah, I like those. Yep. All right, lights are down. I just have to squint to see you guys now, but that's okay. Let's see. Anybody have any other like advantages, disadvantages for either technique? So intracellular, it's kind of nice. You don't have to actually impale a poor neuron. That seems traumatic, and it is. It can be. Other thoughts? Yeah. I think it does. So, yeah, so we have this thing. I want to draw on the board now, and you can't see the board. I'm going to turn the lights on again. Yeah, but they don't illuminate the board. All right, I'm sorry, you just have to deal with me. So the deal with an intracellular electrode, so here's this is supposed to be an axon. So an intracellular electrode, I mean, it's really hard to poke the axons unless you're dealing with, I mean, this is why squid are so awesome, because you can poke actual squid giant axons because they're huge. A lot of times, you know, it's hard to poke, so you have to have this really fine electrode that you can maybe poke into the cell body most of the time. That's usually your best target. Maybe you can poke it kind of near the axon and still get some influence of the axon. So that's hard, but the nice thing about them is that you know, like when you're recording from this intracellular electrode, you know what you're recording. Like you're recording the membrane potential on the inside of that cell, and you know it's the inside of that cell, and you know it is actually the membrane potential that you're monitoring. I mean, it's a more direct measure. So that's a really nice thing about them. Extracellular electrodes, you're like taking a little copper wire, basically, and you're kind of looping it around an axon, or maybe you're looping around a whole collection of axons. That's often what we will end up doing. So you might loop it around the optic nerve, or you might loop it around a nerve out somewhere in somebody's arm. So you have an issue where it's kind of less specific. You don't really know maybe what axons you're recording from with an extracellular. So this is supposed to be extra. You may be recording from a bunch of axons simultaneously. And you're also not like directly measuring memory potential. You're measuring, usually it's kind of like you're measuring changes in electrical activity that are related to changes in memory potential, but it's not like directly monitoring what's going on inside a neuron. So extracellular electrodes, it is a little older technology. Somebody mentioned that last week, and that's true. It's less specific. I think there is one nice thing about them, one benefit of them, and that is that if you're interested in kind of collectively, like what all is the retina seeing? And so if you then take an electrode and loop it around the retina, you kind of get an overall view of the electrical activity going through that optic nerve, because it's, you have, I didn't draw this very well, but usually what you'll have is you'll have your little electrode, and then you'll have like hundreds of axons traveling through that electrode, and you're kind of getting an overall measure of what all those axons are doing simultaneously. Whereas with an intracellular electrode, you're pretty much restricted to one cell at a time. But again, the advantage is you know exactly what that cell is doing, whereas here you really don't know which of these axons is contributing to your signal and in what way. It's a little bit more chaotic of a result. All right, I just like, again, I like to expose you guys to some techniques that are commonly used. Let's do this. So first one, this one, first two on question 2 went to James. So what did you get? If you can kind of like, I don't know, somehow describe, and I'll try to drag. I can't even click on it. Move it. Come on. And I'm clicking in front of people too. It's harder to click in front of people. Get that out of the way. All right. So resting is, you're saying there? Okay. Yeah, fair enough. Let's try to forward enough, right? I mean, we're just trying to get an idea of where these things go. I don't know whose idea that was to. I was like, oh, I'll have them actually manipulate on the word. It's such a pain. Okay, let's do 2 then. So explain the relationship of what is shown in part B, which is here, to what's shown in part C, which is here.
So part B has 3 different voltage-gated sodium channels opening and producing a positive inward current towards the cell membrane. And that's basically summed up in C, where it just shows the net positive effect of all those channels opening and driving the membrane potential towards a more positive current.
Cool. Yes, we're summing that all up down in C. Awesome. So next bit, I'm going to keep going, and we'll go back through if anybody has questions. Explain how part C relates to part A in this figure. So this one went to Emily. I realized I didn't turn the lights down. So it's showing the amount of sodium influx related to that. Yeah. All right. What about, so then part 4 also, wait, yeah, part 4 also went to you. So explain what is shown in part D of the figure. So that's all these little guys, to E. So it's very much like what we're seeing up here. So if C is summing up all of the little individual channels shown in B, E is summing up all the little individual channels shown up in D. Part 5 went to Jen. So explain the relationship between part E and part A. I think that's you. What was the last little thing that leads to the, yeah. And then I think you're also on 6. So 6, this is asking what's shown in part F. So what did you get? Yeah, so you're like taking all those individual sum up ones, and we're kind of, it does label them as red and blue here, but we're just sort of, these are all positive charges, and we're just sort of tracking flow of positive charge. And initially there's this big influx of sodium. And then later we have this big efflux of potassium, and you know, we're basically, if you compare this, it's kind of hard to see it in one screen here. But if you compare the size of the sodium one, it looks smaller than what's above a little bit. It's a little more narrow here versus there. And so what's happening is we're kind of, you know, initially lots of positive sodium is coming in, but then as you start to have potassium going out, you get to kind of a neutral amount of positive charge coming and leaving. And then the potassium takes over and just overwhelms everything as the sodium charge is all shut down, or sodium gates shut down. And then finally, where am I? Here we are. Go scroll down. Last two were about refractory period. So this went to, you go like Tina, right? Yeah. So what'd you get? Okay. And what about the relative one? What'd you put? Anybody put anything, we kind of summed through these already, but for a lot of these, but anybody put anything different or additional for any of these items? Yeah, good, Mary. I'm going to add a little thing. So it's absolutely true that the voltage-gated sodium channels are responsible for what, for the absolute refractory period. They would inactivate right around here. So all the voltage-gated sodium channels get their globular subunit that goes in and plugs them up. And they cannot reset until you get back down to rest. So from here to here, that's basically your absolute refractory period. And so that's the, you know, that is the underlying mechanism behind the voltage-gated sodium channels that results in that. We still have this relative refractory period, though, which goes from the point at which the voltage-gated sodium channels reset. So at that point, they're ready to open. That's starting right there. Until we go through this undershoot and get back to rest way at the end of this graph. So from that point, from the point we hit our resting membrane potential again, all the way through the undershoot till the undershoot resolves, that's the relative refractory period. And so what I'm just going to modify the potassium. So it is the potassium channels that are driving it. I'm going to, instead of closing, I'm going to say being open. So the relative refractory period basically lasts from the point that the absolute refractory period ends to the time that all these voltage-gated voltage-gated potassium channels finally close. So the voltage-gated potassium channels being open are related to the relative refractory period. When the potassium channels finally close at the end of the undershoot, the relative refractory period would then end, right? Because at that point, you're back to rest. It's very easy to open, have another action potential fire because you're not hyperpolarized or anything. We're good to go. I didn't realize we're 2 minutes over, so I have to stop. So sorry about that. We'll pick up on Wednesday.